Oh, cool. So we've started the recording. Okay, perfect. So good morning, good afternoon, and good evening to all, and had a very warm welcome to all the participants joining us today. On behalf of Paleo ECR India and event coordination team, I am 𝒂𝒏𝒂𝒗 along with my co-host today, Gauri. We're delighted to welcome you to another session of Paleo ECR India Saturday Talk series. As I already explained and many times that this Paleo ECR India was established with the simple vision to connect the early career researcher working in the field of different field of paleo sciences and earth sciences, fostering the scientific discussion, collaboration, and exchange of ideas. And we are truly grateful to see researchers, students, and the scientists, associate professors, from different institutes joining us regularly and making, this platform a very vibrant scientific community. Before introducing today's speaker, let me ask you a few questions like, when we see smoke rising from a forest fire, what remains of that events hundreds or even thousands of years later? And how do scientists identify whether that fire occurred naturally due to climate variability or because of some human activities? So the answer to these questions are often hidden within tiny molecular fingerprints preserved in the environment archive, like lake sediments, peat sediments, soils, and marine sediments. So decoding these signals allow us to reconstruct past fire activity and to better understand how the ecosystem and climate have interacted through the time. So to guide us through this fascinating field, we are delighted to welcome today's invited speaker, Dr. Ankit Yadav. So Dr. Yadav is currently an assistant professor at Faculty of Geoscience and Geography, University of Göttingen, Germany. His research lies at the intersection of biogeochemistry, organic geochemistry, biomarker, molecular, fossil, and environmental reconstruction. And through the application of advanced molecular and isotopic techniques, his work helps unravel a complex environmental processes recorded in the natural archives. Talking about his academic journey, Dr. Yadav completed his PhD in Earth Science from IISER Mohali, India in 2022. Following his doctoral studies, he worked as a postdoctoral fellow at Department of Marine Geoscience, University of Haifa, Israel, before joining the University of Göttingen as the assistant professor. His research expertise has a wide range of state-of-art analytical techniques, including GCMS, GCQ, TOFMS, PYGCMS, LCMS, and like many different instrumentation like FTIR, Raman spectroscopy, which enables him to investigate organic compounds and isotopic signature preserved in environmental archives with remarkable precision. So today, Dr. Ankit Yadav will be delivering a talk which is entitled as the Beyond the Smoke, Decoding the Unrevealing Biomass Burning Signature in Environmental Archive. And in this presentation, he will discuss, I think, how molecular tracer and advanced analytical approaches will help identify biomass burning signals. So before we begin, I would like to share a brief announcement that kindly all of you keep your microphones muted through the talk. And please feel free to post your queries in the chat box during the presentation. And we highly encourage active participation during our Q&A discussion session. And this presentation will be approximately 30 to 40 minutes. And then we have one dedicated 15 to 20-minute Q&A session, and I think, which will be moderated by my colleague, Gauri. So with this, it's my great pleasure to invite Dr. Ankit Yadav to begin his presentation. Thank you so much, Ankit, for joining us. And now the stage is yours. Thank you. Now you can share your,
Yeah. I hope it is visible now, right?
Yes, perfect.
Okay. Yeah, thank you, Arvind, for the kind invitation. And thank you for the nice introduction also. But I also kept a single slide to show like how my academic trajectory has been. So I joined IISER Mohali back in 2012 when I was an undergrad student there. So I spent 5 years at IISER Mohali from 2012 to 2017, and then I was in dilemma whether I should continue my PhD here or go outside. So I decided to continue it there, and I joined Dr. Anup Ambali, and we both were very much responsible. We were deciding if we want to go ahead with an organic geochemistry lab or an inorganic geochemistry lab. So that was the first time I had this opportunity of using the instruments, or particularly focusing myself around organic geochemistry. So I'm a trained geochemist, and specifically an organic environmental geochemist. And then after that, I moved to, as I've been told, like to Israel for a brief period of time. Like I spent 4 months there. It was very wonderful time. It's a beautiful country also. And then from there, I moved to Germany. So right now I am teaching students, doing my research, and yeah, spending some time here and there. So without wasting your time, like let's move. So today's talk is a general talk. I would like try to focus on these different attributes from the broader spectra of geochemistry, right? So biogeochemistry, organic geochemistry, analytical geochemistry, isotope geochemistry, environmental geochemistry, aqueous geochemistry. And then currently, like people are going nuts over cosmochemistry, analog geochemistry. So when you use Martian analog, right, extraterrestrial analog. So those are also part of. Have you ever wondered, like perseverance, curiosity? They are not scanning, they are going for the life traces. And organic molecules are those tracers by which you can tell like whether a planet was hospitable for some kind of organisms to grow themselves, right? So that's the beauty of organic geochemistry. I'm not demeaning any other type of geochemistry, but I'm like giving the perks of being an organic geochemist. So you can work around the different domains within the broader geochemistry, right? And specifically molecules. So I try to analyze molecules which are preserved into different type of environmental archives. Sorry? Okay, then I continue. So
Yes.
So we analyze like I analyze specifically these molecules which are preserved into different type of environmental archives. Now, some of you might be working in speleotherms and you would be wondering whether there are any organics in the speleotherms or stalagmites. Yes, there are organics. There are wildfire tracers also in the speleotherms and stalagmites. Now, some of you would be working on some kind of biological organisms. Now, do they somehow ingest these kind of organic molecules, or do they produce these kind of organic molecules? Yes, they do produce these kind of molecules, and they are very much preserved into these different kind of skeletons, which is there into this organisms. So ranging from the biological model organisms all the way to the sediments or rock samples, you do have a plethora of organic molecules. And this is a typical schematic, right? So I kept it simple for the lake sediments. You produce something, any kind of molecule through abiotic processes that gets transported into your water column. Then in the water column, you do have mineral organic aggregates, right? Sediments. It could be soil as well. Have a lot of humic acid present, all kind of other macromolecules present, all kind of other micromolecules present. Now, these things are first absorbed onto the surface. And then over a period of time, this adsorption become absorption. So that's the basic principle how these organic molecules are preserved into the different environmental archives. Now, every environmental archive have certain amount of surface area. Even stalagmites, the calcium carbonate precipitation. So when calcium carbonate solidifies, that's the surface area where all the organics are going to be adsorbed or absorbed. Okay, same in the lake sediments, same in the rock samples, same in the atmospheric aerosols. So whatever you name, whatever the kind of environmental archive you are working on, you have a lot of organics. And they survive from some years all the way to the Archean as well, right? So the earliest traces, what were these archaeas were forming? What kind of organics were getting formed. So that's an evolutionary concept. So if you want to understand about the evolution of organics or evolution of organics, you analyze their organic molecules. And then you make these trees, fancy trees, where everything is getting separated at the later end, right? So yeah, that has been the focus of my work. And within these molecules, we do have some molecules which are very dedicated for fires as well. Now, before going into that detail, let's see how these organic molecules look like. So organic molecules have different functionality, ranging from the very basics. Those are lipids. Now, what does a lipid mean? They are very much abundant into the cellular structure of any organisms. So every kind of molecular organic is not a lipid. A lipid is something which is having a biogenic origin, and in specific, a certain type of origin. You can say the pigments, for example. I cannot say pigments as lipids. They have a very different functioning. Then I have amino acids. I cannot say them lipid biomarkers. So that classification is also very necessary. Every time whenever you are using molecular markers, you should never say like lipid biomarkers. It has to be either biomarkers or molecular tracers or molecular fossils. And then we have lignins, then we have sugars, and quite recently, what we have is non-biogenic molecular tracers as well, which is not present into our natural environment, right? So this is a biphenyl chloride, right? So it is not present. It is present in fire retardants. Okay, so that's of the anthropogenic origin. Then you have these phthalic acid esters. They are present into your plastic material. Then you have these PFOS, perfluoroalkylated substances. They are very much present into different kind of coatings, which are resistant to some kind of heating, which are resistant to some kind of moisture absorption. So all these things. Now they are of anthropogenic origin. They are not present naturally. So you see like where we are heading. We can use the same organic molecules preserved into the environmental archives to understand how much the humans have deteriorated an environment by analyzing these compounds. And the same goes for the contemporary fire events as well, because what kind of fire events you can think of? Catalytic conversion of fossil fuels inside your engines, right? Fossil fuel burning. Then controlled fire regimes. How many of you eat tandoori food? That's also a fire event, right? Then we go do some barbecue also. That's also a fire event. So all those also produce a certain kind of organics. Now that is like what the question Arvind raised. How do I decouple all these things? So before going into that part, and then, yeah, you can use them to understand about the origin of organic material, whatever their implication is. So all that you can cover by analyzing these different kind of molecules. So the very first thing what we do, or what I want to understand, how does a pyrogenic activity generate these organic tracers? Okay, and when they don't generate one classes of compounds. They generate multiple classes of compounds. So there I have shown you, like I took some biomass, which has some lignins, hemicellulose content, some resins, every kind of things are present. Now a combustion event is somewhere starting from a smoldering phase, which is like 200 to 300 degrees Celsius temperature, and all the way to 1000, 1100, 1400, 1600 degrees Celsius, which in ecological term is classified as crown fire. When your conifer trees are burning or any kind of trees burning, when there is an uncontrolled flame, that's called as a crown fire. When it propagates from the low stem to the upper canopy. So that's a crown fire, and the temperature reaches around somewhere around 800 to 1200 degrees Celsius, depending on how much has been burnt. California wildfires, Uttarakhand wildfires. Now there was a recent event in Kasoli. So one of my partner was there, and then he sampled some samples from that Kasoli fire also, how distinct that fire is. Okay, so initially all these macromolecules, which is like these polymeric chains, which is lignin, where you have a varying order of lignin units connected. Then you have hemicellulose, then you have cellulose connected. Now it gets fragmenting. It is getting fragmented when you are increasing the temperature. And when you are increasing the temperature, the polymeric chain is actually getting into monomeric form, dimeric form, trimeric form, and ultimately like these smaller units. Okay, so that's the very first process what happens during a fire event, whenever I'm combusting something of biogenic origin. And ultimately, if I increase the temperature, the overall functionality of that molecule also changes. And that results into these aromatic hydrocarbons. So when you have a lot of free radicals present, reactive oxygen species present, they are responsible to form a different classes of compounds, which are largely classified as polycyclic aromatic compounds, PACs or PAH hydrocarbons. Now that's a high temperature mechanism. So it's basically called as HACA mechanism, where you are abstracting a hydrogen from your parent molecule and you are incorporating an acetylene molecule, which has been formed during this pyrogenic process to form these, different PACs, polycyclic aromatic hydrocarbon. As the name states, they are polycyclic in nature. And the cyclicity increases because of this HACA mechanism, like here. So the very first thing is the hydrogen abstraction, then you are adding this acetylene, and this is getting repeated over and over again. And then you see from a benzene ring, it has all the way gone to a pyrene ring, which is a 4-cyclic aromatic compound. Now, there is nothing biogenic in nature here. All your biogenic stuff is destroyed, is now available in very small molecules. And now that is responsible because of the temperature, the kinetic mechanism, the kinetic reaction pathways is forming a different classes of compounds which are of pyrogenic origin. So that's how the different type of molecules are getting formed inside your entire spectra of temperature range. Okay. Now that's about their formation, the major classification. How do we analyze them? Right? I can tell you 100 stories, but how do you analyze them? How feasible is it to analyze them, and what is the typical process by which we analyze them? So there are broadly, there are two categories. One is called as wet chemistry based analysis, and then ultimately the analytical part of that analysis. In wet chemistry, what we have is a detailed schematic, a framework, what I think 99% of the labs around the world follows. So that's the collection of your samples. You dry them depending on what kind of infrastructure you have. You can use a hot air oven. You can use a freeze dryer. You can also dry them in open air as well. And then you pulverize them because there is a heterogeneity in the samples. You want to homogenize the sample. That's the basic principle behind the pulverization part, or like finely grinding these samples. And then from these samples, we use 3 types of instrumentations: sonication, a sonicator, which would be sitting in one of your corners inside your labs. Then a typical distillation type of technique, which is called a sock slit, where you have a closed chamber. You have organic solvents because organic would dissolve the organic molecules out of your sediments. And then you heat it. And then because of the heating, there is a kinetic energy in the molecules. And when you have a kinetic energy, they would dissolve themselves from the clay surface or whatever the mineral or soil aggregates are there or sediment aggregates are there, from that to a free organic solvent phase. Now that is a time-consuming process. Then we have a very sophisticated one, which are classified as pressurized solvent extractors, or you can also call them accelerated solvent extractors. Now what we do, so this particular machine works on a principle which is a critical point, right? So I have my samples. One way is to use this heating mechanism. What would happen if I increase the temperature also? So temperature is also a function. No, what would happen if I increase the pressure? So temperature is also a function of pressure as well. So ultimately, this machine has 100 bar of pressure and 100 degrees Celsius temperature. So you can imagine what amount of temperature would be inside these stainless steel cells. So if you look at here, these are small stainless steel cells. And then these organic solvents are sitting here. So what we are doing, we are creating a temperature and pressure inside these cells, and we are flushing our organic solvents through these cells. And then whatever you are left with is your total extract, total organic extract. Depending on your solvents, you can either call it as a total lipid extract, you can call it as a total organic extract, you can call it as a total extract. Because if you use a combination of solvents, you can select if you want to extract lipids only, if you want to extract acids only, or if you want to extract pigments only. Okay. And then after that, once we have our extraction done, this extraction has all these different kind of molecules present. So their ratio vary. Maybe 90% of that constituent is lipid, 5% is pigment, 3% is acids, 1% is, let's say, aromatic hydrocarbons. And if I change the solvent composition, that composition of the molecular composition would also change. So now I would have 50%. So if I change DCM, dichloromethane, from 90% to 50%, maybe I'll have less lipids and more aromatics. So I'm just giving you a scenario. And I want to now separate out these things into different classes of compounds. How do we do that? We employ a silica gel column chromatography. It could be a silica, it could be alumina, it could be any other type of these medium. So in typical cases, 95% of the lab, they use silica or alumina. And then if you are very specific for certain analyte, then you use different kind of SP cartridges. Okay. And then we fractionate them. So that's how it looks like. So you do have this column where you see the wonderful color, and you can see that the upper is very dark and the lower is very orangish, and then ultimately yellow, and then translucent yellow, right? So these are different kind of classes of compounds. We classify them into aliphatic, aromatic, and polar. Aliphatic would mean that you just have carbon and hydrogen present. Aromatic would mean that you have double bonds, triple bonds, or slight functionality present. Polar would mean that you have more polar functional groups like NO2, COOH, acids, alcohols, esters, and all these things. So now you decrease the matrix effect inside from your aliquots. I can now take these individual samples to my analytical instruments, and that's the very next thing. So you can use either a chromatographic based techniques, you can also do a spectrophotometric based techniques. I normally employ, or I encourage people to employ mass spectrometric techniques connected to any kind of chromatographic peripherals, mainly GCMS, LCMS, or if you are interested into the stable isotopic composition of these molecules, you can use a GC, which is connected to an IRMS. So this is called as GCC IRMS. And that can give you the stable isotopic, largely known as compound specific isotopic analysis. And then because of the chromatographic peripheral, we do have different compounds eluting at a different interval. And that's how I know that this particular A compound is cholesterol. The H compound is coprestanol. Now, this is a sterol-stenol composition. Coprestanol is present in our feces. So imagine in Holocene sediments, you found out coprestanol, and you are talking about human-induced changes. You can now pinpoint that humans were present. You don't need to dig in for pottery or any kind of archaeobotanical remains. You can simply use these molecules to give you that information as well. And that's how the overall composition now looks like. As I told, aliphatic hydrocarbons, only carbon and hydrogens. Aromatic hydrocarbons, you do have the Huckel's rules or Clar rule following molecular compounds, which are these aromatic structures. Then you have slight functionality in form of oxygen, in form of nitrogen, in form of ester groups. And then ultimately the polar compounds, where you have the good amount of functionality present through oxygen. You have N and oxygen both present. You have these glyceride units, you have acid units. So all those kind of units are present. And that basically is responsible for the polarity of a molecule compared to the non-polar molecules. Okay. And our fire molecules are there into the aromatic fraction and the polar fractions. So one type of molecules, as I stated, that forms at high temperature was polycyclic aromatic hydrocarbons. Now they have a mixed origin. They are also present into fossil fuels. Okay, so they are not categorically biomass tracers. Okay, in the present time scale, but in the past time scale, they can be used. And on the other side, if you are working, let's say, quaternary sediments or quaternary scale environmental archives, these are the biomass burning tracers. Those are monosaccharide anhydrides or anhydrosugars. Now, if you remember that temperature spectral pyrogenic window, which I showed you, I had lignin units and sugar units. I showed you that lignins and sugars form this skeleton inside our biomass. And if you increase the temperature, that polymeric chain is basically fragmented into these monomeric molecules: levoglucosan, mannosan, galactosan. And if you analyze these compounds, you can understand the fire in any kind of landscape. Okay. So, for example, impact of wildfires on Gondwana flora during Permian-Triassic transition. In this one, we analyzed polycyclic aromatic hydrocarbons to understand how much wildfire was there in the southern hemisphere of Gondwana land, because the Siberian traps are somewhere in the north. And if those were responsible, do they had any impact on the southern hemisphere also? Yes, they had a good impact on the southern hemisphere as well. Then we used the same to understand if there is a transition from biomass burning events to more fossil fuel burning events. So because of the humans, there is a constant increase in the fossil fuel consumption. We are burning a lot of fossil fuels, and fossil fuels also produce these polycyclic aromatic hydrocarbons. And that we used into a lake sediments with a wonderful colleague of mine, like Al-Jasil was there, Vishal is there, he was also there in Israel. So we use these molecules to understand about the socio-ecological history. The 𝒃𝒄 movement, this paper was very specific because it traced back the 𝒃𝒄 movement when people started to conserve forests. So the forest burning stopped, and then all of a sudden we had an increase in the fossil fuel consumption derived polycyclic aromatic hydrocarbons. And then quite recently, like the biomass burning trace and the refinement of the biomass burning tracers. That's what we are going to talk about today, like specifically the biomass burning tracers. Now, these are the 4 types of monomers, what you have from these hemicellulose or cellulose units. So this is a hemicellulose unit, and you can see that how different type of units or different kind of isomers are attached to a big backbone. So you have glucose present, you have galactosan present, you have mannose present as well. And if you combust it, that is basically deriving these monomeric molecules. Okay, so this chain gets broken, and this oxygen leaves a H2O molecule. That's why they are named anhydrides, because you are removing a H2O molecule from a parent structure. And that's how this bridged oxygen is getting formed. So the largely classified name is anhydrosugars. There is no water molecule present inside a sugar unit. That's how we name it. And they can be analyzed using a GCMS, GCMS-MS, LCMS, LCMS-MS. And that's what I started. I started their analysis. It involves a little bit of wet chemistry, which is the derivatization agent, because GCMS would not be analyzed these polar molecules because they have this OH, OH, OH molecules. Now, this chromatographic peripheral, those of you have no idea about GCMS, a GC would have a capillary column, which has a silica phase. And this is a polar molecule. And silica is also a polar molecule. And polar would interact with the polar part, and it would retain it. Okay? So it would not leave it inside the gaseous phase and would not let it enter the mass spectrometer of the GCMS. So in order to get rid of that polarity or that hydrogen bonding between silicon oxide and these molecules, we derivatize them. We protect this oxygen group. using something called as BSTFA, a TMS derivative, a trimethylsilyl derivative. And that makes them very much analyzable using a GCMS. And you can, based on the temperature program or your GC, you can elucidate. Like here, I can show you that the first eluting peak is galactosan, then a mannosan, then an internal standard, which is like D7 deuterated levoglucosan, and then finally levoglucosan. Now, how do I know which compound belongs to which of the peaks? Based on their mass spectra. You can see that 73 is more abundant in the D7 levoglucosan compared to the 206 and 220. If you come here and if you differentiate levoglucosan, you have 204, 73, 147. And if you compare the overall intensity of these ions, we can specify which molecule corresponds to what kind of spectra. And then like galactosan, the 217 is the most dominant one compared to the 73. So you see these reversal of ions. Now the overall, the ions are same. Why? Because they are stereoisomers. They have the same structural orientation, but they're stereoselective, which means that one carbon is oriented into a different direction than the other carbon. So that's why their ions are more or less the same, but their relative intensity is very different. And that's how we analyze these molecules. Now, there is a problem with this analysis, with this particular approach, which is this BSTFA derivatization of anhydrous sugars. This is quite sensitive. If you complete your reaction, if you don't analyze in the next minute, the loss of TMS would happen. And ultimately, it would get converted into from 3 TMS group to 2 TMS group. And then you have 1 oxygen and hydrogen again present. And in most of these environmental samples, we do also have a lot of reactive oxygen species as well, which acts as a catalyst when we store our aliquots inside fridge or inside any kind of like storage place. These reactive oxygen species attack these bridged oxygen and would open this. And then you will have this glucose pyranose. So you are basically changing the overall characteristic of a molecule which has a certain source to a different type of molecule. Now that's the downside of GCMS-based analysis, and in specific, this TMS derivatives of these biomass burning tracers. Now, how do I correct it? One way to correct is if I analyze it in its raw form, which would mean that I analyze OH, 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 OH when all the oxygen groups are present in their pristine form and I don't derivatize them. How do I do that? I employ another analysis, which is LCMS, liquid chromatography mass spectrometer. Now I started digging in if there are any techniques or if there are any methods which have employed LCMS to characterize these molecules, polar molecules. So that is a basic difference between a GCMS and an LCMS. When you have a good amount of functionality or polarity present, we go for liquid chromatography. When your molecules are volatile, we go for GCMS. Okay, so that's the basic characteristic. Now I have polar molecules. They have a good functionality. They are very polar. So I looked for some methods and I found out 2 methods. One was Hofmann's, which was published in 2013, and the another one was Schroeder from the same lab. Now, if you know Hofmann, so she is like one of the pioneering person who is developing methods in characterizing or in isolating these molecules from the environmental samples. And I followed her method. Now, she is well known, okay? Maybe her method is also correct, and that's what we started. So that was the very first thing I did. I tried to analyze the mass spectra of these 3 isomers using a LCMS-MS. And I found out that all the 3 isomers have a distinct mass fragment. For example, levoglucosan has a 161 to 113. These 2 are very much abundant. So 113 is present into this one, 161 is also present. But if you go to mannosan, 113 is not present. So that's a distinct mass. I can use this mass to analyze my molecule, okay? Now I use liquid chromatograph to elucidate it. Now it's just like explaining these 2 cups I have two identical cups, their color is different, but if I break them, their fragmentation would not be equal. And that's actually the character of every molecule as well. When you break them, they produce distinct ions. Even though they are structurally very coherent, they are stereoselective, which means that your cup handle would be very different from the another cup handle. That could be color, that could be the size of that broken fragment. And that's the basic principle what we work on. Now, when I did that and I dig in through their literature, I found out that that is the elution pattern which they reported. Galactosan is eluting priorly, then mannosan, then levoglucosan. Okay, they could able to disentangle that part, but not in my work. Okay, so that was the major finding of this work. I reported that mannosan is eluting first, then galactosan, and then finally levoglucosan. So that part is called as isomeric assignment. So based on my studies, I stated that this person has isomerically misassigned these isomers. Now, why this is important? Because there are certain ratios based on these biomass burning tracers, which you use to understand what was burning. Now, imagine Uttarakhand, you have a lot of conifers. Conifers are largely classified as softwood. Softwood has a lot of mannosan and galactosan. Okay. Then, if I move to grasses, the overall abundance of these isomers would decrease. What would happen? Now, if I am burning grasses, if I'm burning conifers, if I'm burning hardwoods, if I use the ratios or overall changes in their composition, I can understand what was getting burnt, whether there was a conifer proliferation, whether there was like these terrestrial trees, deciduous trees which were growing, whether there was an expansion of grasses. So by analyzing these isomers, that is very much required. Okay, and that's the major finding because that would ultimately define these ratios. And we further tested that into a mixture because initially I tested them into their individual state. Now you can, like a person who is an analytical person also would say that maybe when you mix them, their elution would change. No, that was also not the case. In my mixture also, I found out that my mannosan is eluting prior to galactosan and then finally levoglucosan. So completely reversing the elution order of someone who is very well known in the field of organic geochemistry. And that's what I mean to say that you need to question and validate your methods and not blindly those methods. So you need to bring more understanding about the processes, whether that's analytical, whether that's environmental based, in order to bring something new to the community. Okay. And then what we did, like I told you, we had our levoglucosan to mannosan ratio. We have our mannosan to galactosan ratio. And I did a simple kinetic experiment. I derivatized my samples. I left it for 6 months inside a minus 20 degrees Celsius because I was worried if the TMS derivatives, which we did for GCMS analysis, would hold their character or not. And this is what I found. So if you see this one, that T is equal to 0 days, my overall ratios for GCMS and LCMS are more or less the same. They are equal, which means that my actual character of molecule is still present. But what happened after T is equal to 180 days? My GCMS ratios changed a lot because my parent molecule is not present now. And my LCMS, which is underivatized form, is still holding its character because I didn't play around with the molecule. I simply left it in its parent state. Okay, so if you are employing certain kind of this wet chemistry based methods, you need to be careful what you are doing and what kind of repercussions it can have on your data. And this is that scatter plot, what we use largely to see what is the combustion source, where they are getting derived from. And depending on your analysis window, time window, you can see that there is a greater diversion between these source attributes. So Yeah, that's all. And this is just a simple chromatogram illustrating like pH also, which you can also use to understand about the wildfires. So this was all for today. And with this, I thank you guys.
Thanks, Ankit. I think, Gauri, can you please take care of this Q&A session?
Yep. Am I audible?
Oh, yes.
Okay. So thank you very much, Dr. Yadav, for that insightful and engaging presentation. So your talk provided a fascinating overview of how careful molecular characterization can improve our understanding of biomass burning records and paleoenvironmental reconstructions. So I would request all the participants to type in your questions in the chat, and then we can take it one by one.
Mm-hmm.
Or they can directly raise the hand and ask the question. Even just unmute yourself and discuss.
We have a question in the chat by Madhav. Why addressing matrix effect is important?
Okay. So metrics can be seen in two ways. One is like when you have a total extract. If you analyze a total extract that has a different amount of metrics, because every kind of molecule is present, ranging from non-polar all the way to polar and macromolecules also. So first thing, this matrix effect plays a role around your concentrations. Now imagine I have two samples. One is from late Holocene, one is from very young samples. From late Holocene, my TOC was close to, let's say, 10%. And also in my younger sediments, my TOC was also 10%. Now I have the same TOC. What it would mean? It would mean that more or less my organic content is same, right? That's not the case, because you have labile organic matter also, and you have recalcitrant organic matter also. So maybe in your late Holocene samples, your recalcitrant organic material is still present. And if you are going to quantify in the overall organic matter inside that particular one using any kind of chromatographic or mass spectrometric techniques, the other molecules would play a role in deciding the concentration of this molecule. So I want to get rid of maximum metrics from my analytes aliquot and have to have much more precise concentration, because that would determine like how do you interpret your records in that particular scenario. I just gave you a Holocene scenario. And yeah, in the current context, it would determine like how much of the carcinogenic pH are present into your atmospheric particulates, let's say. So if you report, let's say 600 nanogram, and after correcting for the matrix or after removing the matrix, the concentration can go up to 800 nanograms. And that's above the threshold limit. So that's why the matrix effect correction or matrix, removing the matrix from your analytes overall aliquot mixture is really important. I hope I answered it. Madhav.
We have another question by Anant. I have a question regarding the storage and time constraint before the analysis of molecular signatures. And if one has kept the sample for a longer time and dried the sample in an oven for at 40 degrees, can these samples be used or not for the analysis of molecular signatures?
Yeah, so that's the major problem. So deciding on what you are analyzing, your initial drying method is decided. So for example, if I am analyzing something which is a semi-volatile, what does semi-volatile would mean? That if you leave it open, maybe most of you are using naphthalene bowls inside your houses, right? To store your clothes or to prevent something from, say, any kind of insects and all these things. Now, naphthalene is a polycyclic aromatic hydrocarbons. When you keep these naphthalene bowls inside, it evaporates. That's the semi-volatile nature. So if I have something which I'm drying over 40 degrees, I'm making sure that many of my organics are leaving that sample. Okay, but that is for the quantifiable part. But if you are doing something, let's say isotope ratios, there I don't need to worry about 40 degrees or 60 degrees or 80 degrees, because there I am looking at intrinsic character of a molecule and not the overall abundance of that molecule. So if you are working something related to the compound specific isotopes, you can use that much very well at 60 degrees Celsius. So if analyzing the copperestanol signature, yeah. So if you analyze copperestanol signature, now copperestanol has an oxygen OH group, which is connected to an ester, right? And before analyzing copperestanol, we do deesterification process also. So if you somehow play around this moisture availability and remove the moisture, that can further condensate more of the free copperestanol into these esterification things. Okay, so I would not suggest. I would still suggest freeze drying, because I want maximum of the free molecules and not in the esterified forms, so that they can be like, in worst case scenario, I can analyze free copperestanol signatures and not the esterified copperestanol signatures. I hope I answered it.
I hope as well. And we have one more question from Madhav. What can be the best way to quantify soot or pyrogenic particle? Like, different methods have their pros and cons.
Yeah, so this is something I'm being developing right now. So soot, if I go to that particular slide, I hope I have it. Yes. So soot is something which is like 1000 degrees Celsius. How much of you know like about fullerene? This is this ball type of structure. That's a soot. That's an elemental carbon. You do have these carbon, only carbon and hydrogen present into these big molecules. Those are soot. Now, if you are specific about the soot, we go for either a ramped pyrolysis, which is like TC analysis, but I combust at 100-degree window. So I do a ramping of 100 degrees, keep my organic matter burning at 100 degrees, then 200 degrees, then 300 degrees, and then all the way. Now, that produces certain amount of CO2. Now, that CO2, if you categorically quantify, that gives you that window. Okay, 800 to 1000 degrees Celsius, if I combust something, that would be my soot signatures. If you want to do something molecular level, you need to digest your soot to make it BPCA, so benzene polycarboxylic acids. Now, I didn't cover that part here, but those are these, they are derived when you break these big soot molecules into micromolecules, like really micromolecules. And if you quantify these BPCAs, you determine what amount of soot is present. Overall, if you are talking about pyrogenic carbon, you need to integrate each and every type of pyrogenic molecules, ranging from lignins, sugars, aromatic hydrocarbons, acids also, then finally BPCAs also. So that's the best way. And if you want a broader quantifiable amount, you go for this ramped pyrolysis. That's the best way to quantify the pyrogenic carbon from any kind of environmental metrics. Yeah, I think, Madhav, I answered it. Or do you have anything else?
I think I'll move on to the next question by Ajay. Is it possible to distinguish the degree of fire using these proxies?
Oh, for sure, for sure. So one of the projects right now I'm working are around some of the Neanderthal settlements. Now, when did Neanderthals start using fires? So when you combust something, do you combust it like 600 to 1000 degrees Celsius range? Or would you combust it around 200 to 300 or 400 degrees Celsius range? Like if you are doing barbecue, what is the temperature range for a barbecue? You use oven in your houses, 220 degrees Celsius, right? So that's the humans made fire, right? And if you have wildfires, that is uncontrolled fire regime. Then the temperature would go 600, 800. But if there is something of human origin or controlled fire regime, you would have that signature. So that is the example I took. like controlled fire, which is 200 to 300 degrees Celsius, uncontrolled fire, which is 600 to 800 degrees Celsius or 1000 degrees Celsius. You can very much distinguish using these proxies.
We have one more question from Madhav. Is there any exact temperature of soot formation particle?
Is there an exact temperature of soot formation particle? Not really. So if you control oxygen, your soot formation would happen much more quickly. You need a critical temperature, like it has to be above a certain temperature. I don't recall that temperature, but it is in the same window, like from above than 600 degrees Celsius for sure. But categorically, what is the exact temperature around which so it really depends on this mechanism. So this mechanism, you can see that this is a polycyclic aromatic hydrocarbons. That's a small molecule. But if I further add 4 more rings, or 4 more rings, or 4 more rings, if I make it 16 carbon, 16 cyclic group, that becomes a soot. Okay, so that determines if you do not have any oxygen. So if you have an oxygen or nitrogen or any kind of these other atoms present, they would incorporate into that particular carbon skeleton. But if not, you have a oxygen limitation, no oxygen present, only temperature present, then that would succeed in forming more heavy molecular weight soutes. So the temperature is around, I think, 800, somewhere above that one, for sure.
Thank you. And we have another question from Nitin. Do you encounter post-depositional sediment diagenesis effects, marine or lake records, that overall affect the organic matter? Is it a blessing or curse for paleoenvironmental records when using organic geochemical proxies?
Mm-hmm. Yeah, so that's the one problem we have. So as soon as the biological organism or any kind of biotic stuff is dying, the degradation happens. Now it really depends what kind of analyte you are using. So for example, if I have, so let's say lipids, they are very much preserved. They also degrade, but their rate of degradation is much more slower than sugars or amino acids. So it really depends like what you want to analyze. So if you want to just analyze the carbon skeleton and the hydrogen skeleton, which is these aliphatic hydrocarbons, you don't need to worry about the degradation, because your degradation would be very much the same throughout the core or throughout the environmental record. But if you change your analytes, you really need to be careful, and you need to supplement it using the other kind of indices. So one of the common indices what I use for the lake sediments would be CPI, carbon preference index. So if I have a carbon preference index equal to 1, I state that my organic matter is totally matured. It is of no use for understanding something, but it can still give me many informations. So it's kind of a blessing, I would say, because still I showed you like end Permian Triassic extinction, right? End Permian extinction. We analyzed pH. We could decouple the wildfire signatures that were there in the end Permian. It was not just floods, it was wildfires, and that too in the southern hemisphere. I don't know how much of that Siberian trap feedback was responsible for this part, but there were wildfires. And also the same kind of molecules, vegetation-specific molecules. So early Triassic is considered to be the evolutionary time frame for the conifers. Okay. Now, I do have some molecules in specific retin that can tell me, and that we pointed it out. We had no retin in the end Permian, but we had retin present into our early Triassic, which told us that, okay, conifers were evolving during early Triassic. So that's a blessing, right? Maybe I could also support it. Initially, it was getting supported from your these macrofossil remains. Now there are molecular investigations also, which also supports that, okay, trees were evolving in a way that they were producing resins. And this conifers have a lot of resin, and retin is a byproduct of these resins. So I can evolutionary. So if you are working on some evolutionary concept, these molecules are a blessing.
Thank you. And now we'll move to Arvind.
Mhm.
First question. Yeah.
So, thanks, Ankit. It was a nice talk. I am actually not an organic geochemist. Basically, we have to follow this non-traditional isotopes. Just everyone, general question maybe like, can we use this paleophile record to identify this abrupt climatic change, like younger dryas and HS, like Henry Stadel time, monsoon shift. Because if you talk about the younger dryas, it's a general theory right now that is one kind of a prominent intense cold event after the last deglaciation, and just like a other DO and HS cycle. So now the study is coming and is showing that this cold event was because of such kind of meteoritic impact. So the huge dust came into the atmosphere, and then it stopped the sunlight coming from the top to the surface. So, but still, this just one or two studies are coming. So that's what, like, can these records be used in the later phase to identify this kind of abrupt climatic events?
Oh
This
yes,
paleophile record.
oh yes. So one of the projects I'm working on is ICDP project, which is around the same objective. That's the deglacial, glacial, and interglacial periods, and how has the fire evolved throughout this phase. So the ICDP is from Lake Melville, which is next to Canadian coast. And we have cores. I analyze these samples. We also present into one of the ICDP meetings recently. That was in somewhere in Germany, I forgot. And we very much found out that when during glaciation, we did not had any fire, of course, and that too from the Western continent, this northernmost continental part. But during interglacial and the deglacial period, I see an increase. Okay, so and then afterwards, like in the younger ones, we do have an ample amount of fire events. So yeah, so this is what we saw, and we also witnessed a lot of charcoal also. And so, for example, many times it's also the case how much smoldering phase, right? It could be a smoldering phase fire as well. Now, smoldering phase would not produce charcoal. If you are producing charcoal, you have to have above a certain temperature range. Now imagine if there was smoldering phase fire, like localized fires, like a cluster of fires, but at very low temperature, I would not have any charcoal. And the concept would be, oh, there are no charcoal, there were no fires. But let's now dig in through the molecules. That would give you this information that smoldering phase, which is relatively less temperature combustion conditions, would produce these molecules, which are characteristic of this certain low temperature pyrogenic activities. And that's what we are proposing into this this project. That's ICDP project. Yeah.
Just one more query, like being a paleoceanographer, like we are very much familiar with that. We use this for geochemistry or isotope proxies for pilot temperature reconstruction. But I have read one or two papers regarding the use of this liquid biomarkers for reconstructing the past temperature in the marine sediments. I just read one paper from, I think there is one lady from CNRS. So like is it possible to do it, because people are doing like they're trying to reconstruct the past temperature, but that what are the limitations if you compare this proxy with a traditional geochemical proxy in estimating the past temperature, because it's still a
evolving
huge
thing.
risk
Yeah.
actually. Yeah.
So the molecules which you are talking about are called as GDGTs, glycerol dialkyl tetraethers. Now, let me see if I have that figure here. Maybe I have somewhere. Just a second, just to give you a context. Yeah, so this is something like a glycerol dialkyl tetraethers. Now, if you see that carbon is attached to an oxygen, carbon is attached to an oxygen, this carbon is also attached to an oxygen. Okay. There's something called as catenation power. So catenation power would mean that the carbon-carbon bond, why does carbon accumulate and not oxygen, or not CO? Because their bond strength is not that much that could survive a certain amount of diagenesis, right? So if I have alkanes or acyclic isopenoids or terpenoids, they would survive much more longer. I can trace these aliphatics to even Archean samples as well, but not GDGTs. They are highly going to, let's say, quaternary. Their applicability is till quaternary. The largely tested. Some reports claim that they can also go to Paleocene as well. But so the very idea is like, because of this functionality, the oxygen thing. And what would happen, that this bond would break. And if you check the other skeleton, like this skeleton here, that's a carbon, 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 carbon. So your parent structure would be lost. But this functional unit, this carbon unit still will be present. So you can say that, okay, these temperature indicating molecules are still present, but not in its pristine form. Okay, they have lost its character. Now I cannot use them to reconstruct temperature, and would either need to go to delta 18O or any other kind of inorganic elements. So that's the limitation for this typical classes of compounds. Now, for example, lignites, like lignite is a coal. Lignite have a lot of sugar still present. So you can analyze like these sugar units into the lignite deposits as well, but not GDGTs, because they can go under a great amount of degradation. They are very useful for the Holocene, Quaternary, very, very much useful, even in the marine, even in the terrestrial archives. In the terrestrial archive, they are a little complicated, but in the marine, they are very much there because of the extremophiles. Yeah,
Thanks.
so I think there is one more question. Two more questions.
Okay, yeah, we have more questions coming in. From Anand, the question is, what are the sampling and storage methods for the analysis of copristinol in leak sediments?
Okay. So if you are working on copristinol, I would ask you to freeze dry them first, store them at -4 degrees Celsius as soon as you have sampled them, not to subject them under oxic conditions, because as soon as you subject them under oxic conditions, that would initiate the microbial aerobic degradation. And what would happen under aerobic degradation would be that your copristinol signatures would be lost. That would convert into a copra. That's a kind of a double bond ketone. Okay, so your OH would convert into a double bond O. And freeze drying would help you to not make more esterified copristinols and would have much more free copristinol inside your sediments. So that's the basic advice I can only give you. That is like to store them at -4. Simply, as soon as you take them out for the sampling, just freeze dry them, and thereafter, like, everything is gone. Yeah.
So we'll take the last question from Madhav, and then we can call it a day, I guess. What is the major problem which is faced or can be taken care by a researcher while using biomarkers as a past wildlife tracer?
What is the major problem which is faced or can be taken care by a researcher while using biomarker as a past wildfire tracers? So the very first thing would be like not to speculate anything, I would say. Just by analyzing one class of compounds, I cannot say that they were wildfires. So I told you guys that pH are also present into your petroleum settings. So for example, in coal, okay, let's say you didn't find any charcoal, but there is a coal deposit. And coal also have a lot of polycyclic aromatic hydrocarbons. So by just not analyzing polycyclic aromatic hydrocarbons, I cannot say like they were wildfires. I have to be extra cautious. I have to take them, normalize them to the overall organic content. I have to trace back to the other classes of compounds also to see if there is any selective partitioning between the different kind of molecules. So that is the one thing I think there. And on a broader scale, so if you have, let's say, a lot of high temperature combustion events, you will not find these molecular traces. You will mostly find soot then. And then your question comes like, then you need to analyze soot also, not just molecules, but also soot. And there are two ways by which you can say that how much of the soot was present. One is this ramped pyrolysis, and the another one is to further digest that into smaller molecular fragments. So those kind of like having a complete picture is very much necessity, not just like sticking to one single indicator. In terms of I'm talking about the paleo records, but in the present record, like those things are like very much observable also, so that you can rectify your observations using any kind of modern techniques as well. Yeah, I think was it fine, Madhav? Okay.
So I think because of the time constraints, we'll have to end the talk here. I would like to thank Dr. Yadav once again for this engaging presentation, and I wish you all the best for your future projects. And thank you for all the participants for joining in and for your active participation. And we'll meet in the next talk if Arvind has to add something else, or if anybody has any other comments.
Yeah, so just I think thanks to Ankit, and actually I was in touch with him, and we are facing the time, this bit timing. But finally, he got some time for us to give this, because I read his recent paper and I was very much fascinating about the paper. Okay, let's discuss something about this organic geochemistry process. So I think I love this talk, and for me now, because I'm writing one proposal for my next postdoc regarding the lipid biomarker. So it's like this talk is very much helpful for me. And so thank you so much all. Thanks to all, and thanks Ankit for joining us today. We will upload this talk on YouTube, and then you can, whoever is not present or want to recheck all the things, can go there and watch the talk. Thanks everyone.
Okay.
See you. Bye bye.
Yeah, thank you, Arvind. Thank you, others. Bye bye.